We'll get going. Tradition in Australia is to acknowledge our traditional owners. So I'd like to acknowledge the traditional custodians of the land where we work, live. And up here on the Sunshine Coast, it's the Gubbi Gubbi people. And we acknowledge their spiritual, cultural belonging and recognise their continuing connection to land, waters and culture. And we pay respects to their elders past, present and emerging. And with that, I would like to introduce Brian Pfister, who is the Vice President Global Medical Affairs of Praxis Precision Medicine. And he's going to talk us through a new investigational drug that they're working on at Praxis. He's going to discuss how it's different and how we're evolving in our anti-seizure medications and how this treatment can be a potential treatment for either our 2A or 8A children. So with that, I'll hand over to you, Brian.
Excellent. Thank you. I appreciate it. I appreciate the opportunity to be here. For us, it's very exciting. For me, it's very personally exciting. I think as a lot of people, may know, I have a son with epilepsy. So in addition to working on different medications for other people's children, this hits very close to home for me. And my child has a little bit different than SCN2A variant, but we understand some of the struggles and have lived some of those as well. So anything I can do to make your lives better is where I'd like to be and what I'd like to do. So I'll jump into some slides to illustrate exactly what we're doing, so you can get a better feel for what we're trying to accomplish. So we'll talk today about Relutragine. So Relutragine is the non-marketed name of the product. So it is not yet on the market, still investigational use. So the data that I'll show you here are from our top line EMBOLD phase 2 trial. So just to keep that in mind, it is not commercially yet available, but we're trying to advance that as quickly as possible to change lives as much as we can. So when we look at it, I always like to start out with the mission. Like, as I mentioned, my son has epilepsy, so this is very near and dear to my heart. But as a company, and I think this is what separates us from a lot of other companies out there, is we really want to help patients by delivering life-altering treatments faster and more effectively than has been done before, and do it again and again. So as you see, we have one of the largest pipelines in genetic epilepsies and also then general and focalized epilepsies. So what this means to us is this is really what we're trying to do is do things that other people have not done in places that other people may not have tried to push the envelope. So we're very innovative and we're very willing to look at things differently and to try different things in order to accelerate treatments to market for patients. So I just wanted to show you just in the general landscape of how drugs are developed and why this is important. So you have a series of phases, and people may have heard the term phase 1, phase 2, phase 3, those type of things thrown around. You can look at this schematic and you can see from early discovery, then through preclinical studies, then into phase 1, 2, and 3. These are the clinical trials where it's in humans. And then after the end of phase 3, then we can get approval for this to be more broadly available to patients and families. But what I wanted to highlight is while there are a lot of steps and a lot of hurdles along the way, there's a lot of opportunity. Each one of these chevrons represents an opportunity for us to push and cut time off the development process, which ultimately is, I think, critical to families to get new medications. A lot of the medications being used today are ones that have been around for 30, 40, 50 years in some cases. And so the idea is how do we do this faster and better? And how do we do it in a way that's safe and doesn't compromise patient safety in the long run? So we look at this and looking at SCN2A, one of the other components I think that sometimes gets lost is actually in the genesis of the company at Praxis. We do have co-founders who have children with SCN2A mutations. So this is something where we look at, why does a company do what they do? We do it out of the passion for the patient. And we do it in a way that we believe has a sense of necessity. We need to go faster. We need to do this in a way to get better medications available more quickly because it does mean a lot to the families. And we believe and know in our hearts of hearts that we have probably the best technology and the best molecules, out and available to make those differences for the families. And again, we try to push this innovation piece, and we try to do it as quickly as we can. So when we look at this, and I'll talk about in these first sections here, we'll focus on early discovery, preclinical studies, and phase one, because each step along the way, we get evidence that'll help us to support a decision. And so when you look at the early discovery, we're trying to identify molecules. As people have mentioned in the past, we look at how is the mutation or how is the variant in SCN2A changing the way that the sodium channels are functioning, and then does this give us a clue and an idea about how we can address this underlying need more effectively, more precisely, and in a way that hasn't really been done before. So that's where early discovery plays a role. And in preclinical study, we can test these hypotheses before we ever go into patients. So what's important to note is these drugs have been studied for a long time before they do make it into patients. And then what we do is in phase one, go into the healthy volunteers who don't have a channelopathy, who aren't experiencing epilepsy or any type of seizure activity. And then we can kind of see how they respond to it, because they're going to be much more sensitive to a downregulation or a change in that sodium channel. So this gives us a lot of understanding into the molecule well before it ever reaches the patients and the children. So when we look at relutragine, what's different and what's unique about this is the functional state modulator. And I'll show you on the next slide what that means. But the functional state modulator is different than a sodium channel blocker. We've heard the term sodium channel blockers in the past, and those typically affect the sodium channels and do exactly what the name says. They block the channel. This one will be more selective. So relutragine is going to be more selective and more precise in its activity, which is going to be a net benefit to the families and to the patients in the long run. What we do see too is the initial data signals to us that in addition to the potential for seizure control, there does seem to be this opportunity for disease modifying. And I'll show you that here in a few minutes. But for the SCN2A community, I think this signals a huge, huge opportunity and really elevates, the level of importance of the next phases of development to get these drugs on the market to help the multitude of patients who really could use better, newer treatments. So as mentioned, I'll take this diagram. I've used this a number of times. I do like this. It's less scary than it looks, and it's much easier to understand. So I'll help you. I'll help you understand what this looks like and what is happening. Functionally, when I talk about the idea of a functionally selective modulator, we look at sodium channels, and you can see here these are the normal activities of sodium channels. So you have the closed channel, you have an open channel, and you have an inactivated channel. And we know that the sodium signals activity in these channels and really is what we've targeted for a number of decades, trying to block these channels. But what we've figured out and what we've understood within our research at Praxis is that we don't want to block every channel. So the normal channels, we really don't want to block those, because what that contributes to is the side effect and tolerability that we see with some of these sodium channel drugs. So we want to leave these channels alone. So what we need to do is create a molecule that is much more selective and much more potent to address this issue here, where we have this variant that's causing a pathological channel function or an abnormal channel function. So typically what we see here is this channel never really gets into this inactivation or resting stage. So it's always open, always ready to fire, always ready to signal. And so then the sodium ions, the sodium that is able to pass through, is able to trigger seizures at relatively low concentrations. So what this means is a lot of seizures. It means patients tend to be more susceptible to drug level changes, or may require more drug than what would typically be used in, say, a focal patient. So what this means is when we can more effectively target this, leave these alone, let them function normally, the net benefit will be reduction in seizures, but should also be lack of sedation of the patient. Because we've seen sometimes when you block channels and block these normal functions, the patient will kind of be sleepy or groggy, or may not be as interactive in the environment around them. And so we know that that's, as a parent, that's definitely not what you want. You would like to have the seizures go away, but you would like to have your child present and be able to engage in the environment and really have a better outcome. So what we've developed is a molecule that will actually do this. And so what we see is, so again, a few more kind of complicated looking graphs, but what I want to show is where this is different than the other drugs that have been used. So drugs like lamotrigine or carbamazepine have been used as sodium channel blockers. And you can see this black line. Each one of the molecules has a black line on it. And this is the persistent current. This is where it's engaging as I dose the drug higher, then it'll have different properties. And so what we see is with lamotrigine, we have to get to high concentrations to get control of the persistent current. Same thing with carbamazepine. But when we look at Prax 562 or Relutragine, our drug, we can get much earlier engagement and control of the persistent sodium current, which is contributing to a lot of the seizures that children have. So when you look at this, and there's this heavy red line in each of one of these, this is typically where the side effects kick in. So what we've seen is historically things like lamotrigine or carbamazepine have to be dosed up at high levels, but they also get into this tonic block, or they get into this side effect causing concentration. So the space between, if you want to think about it in a kind of a simpler term, the space between the black line and the red line, or the blue line and the red line indicates, the potency and the likelihood of having side effects. So what we see with Prax 562 is both the blue line and the black line are engaged well before you would engage this red line. So ultimately, this is where we see the decreased likelihood of side effects and decreased likelihood of tolerability. And when we look at the mouse models and we look at the way this molecule functions, we can get usually 15-fold higher, so 15 times higher the concentration that would be therapeutic before we would run into this red line. So this signals a huge benefit for the families, because usually polypharmacy, like you have seen patients who have been on both carbamazepine and lamotrigine, a lot of times the polypharmacy you see with sodium channel drugs is required because this red line and blue line are so close together. And so then this is what happens is I have to give you another drug and give you a little bit more of it to help try to control the seizures. And a lot of times it doesn't work as effectively. And then I end up with many more side effects. So that's kind of where these are different. And this is how we know the drug is different. So we designed it to be different. We've seen it in the way it functions with the normal channels. It's different. And now we see it as compared to other molecules, that the concentration and engagement and the effect is going to be different. So this allows us to move into, the healthy volunteer studies. And we want to look at what happens, as I mentioned, in this highly susceptible patient population, like the patients who don't, or these healthy volunteers in this case, who don't have epilepsy, are going to be more susceptible to side effects than patients who do have epilepsy, because these would block the channels in a way that it wouldn't be normal for a healthy volunteer, so to speak. So we looked at this in 130 volunteers. And so they were basically patients or participants who came in and said, yes, I'll take this drug. We'll see what happens. What we saw, and what to me signals what I've not seen before in sodium channel drugs, is we didn't reach a maximum tolerated dose. So MTD is a maximum tolerated dose. So if you think about it, it's kind of like tapping out. If I'm taking a medication and I get to a point, I'm like, man, I can't tolerate this anymore, I got to stop. They would be tapping out. And that's a maximum tolerated dose. And what we saw with this is the patients, or these healthy volunteers who don't actually have epilepsy, could continue to take the drug at higher levels, well above where it would be therapeutic. So this is something that most of the time with sodium channel drugs, the patients or these healthy volunteers would feel groggy, confused, might have issues with gait. They wouldn't be able to walk normally, kind of feel drunk, if you want to think about it like that, because it's sedating the system and suppressing the system. We didn't see that with this drug, and this is the first time I haven't seen that with a sodium channel drug. So this signals to me that everything else in the preclinical and discovery is falling in line with what we would expect to see here. And so one of the things that then comes into question is, are we getting target engagement? Creating medications and medicines for to treat the brain are very difficult. So then when you're not seeing some side effects that you thought you might see, then you go and you look at biomarkers. And sure enough, we did see that the drug was actually having an effect, and that the medicine was having an effect. So this tells us we're in the target organ, we're in the brain, we're doing what we expect to do, but we're doing it in a way where we're not making the patients incredibly sick. So this is, to me, one of those goosebump moments, because as a researcher, as a developer, we've been trying to do this for decades, and now we're starting to see this come to fruition here within the Relutragine 562 program. So when you get through the healthy volunteers, each step of the way, as we get over those hurdles of drug development, we get over those challenges that typically come up, it allows us to move into the next phases of development and allows us to have discussions with regulatory agencies. So what this means is, as the healthy volunteers did well with the drug, this allows us to be confident going into the phase 2 trial of SCN2A and 8A patients. That's the EMBOLD clinical trial. We're able to go into that study and really look at this now in the disease state or in the condition that we're most interested in, which is the 2A and 8A populations. And we want to see, do we see this effect? Are we able to control the seizures? And are we able to give something else beyond just seizure control? So this is where the story kind of takes a little bit of a pivot. So typically, clinical trials have to be done in the clinical trial office. You would go to the hospital, you would be in the office for a number of hours. When we looked at this and we looked at the SCN2A and 8A patients and families, one thing that stood out to us is that really some of the patients who would be most likely to benefit from the product sometimes can't be in the clinical trials because it's too hard to travel. It's difficult for patients to travel long distances and families to pack up and go. So we wanted to come up with a better way of doing it. So this kind of gets back into the innovative idea of Praxis and what we're trying to do. So we looked at it and said, well, is there a way that we could do it at home? Is there a way that the children can stay home, the families can stay home, do it on their schedule in the comfort of their own home in a way that doesn't create a lot more difficulty for them to participate? So we did find a way to do a DCT or decentralized trial, and that basically means that there's no central site that has to be traveled to, but they can do it at home. And so what it allows us to do is day one, the patients will have a clinician, so a physician will come into their house. They'll do the normal things like a physical exam or neurological exam. They'll look at the med history and concomitant medications. And then they'll draw some blood to look at, are the baseline vital signs and baseline function okay for the patient to continue into the trial? So at that point in time, they'll start counting seizures to see how many seizures they're having in a 28-day period, because ultimately it will start from when they're untreated or not treated with the investigational product, and it will look at the end when they have been treated with the investigational product and to determine if the effect that we're seeing is related to the drug or not. So everything's done at home. The nurse and physician come back on a monthly visit to check on the patient, and every 2 weeks in between to call to check on the patient. So there's a lot of monitoring and additional monitoring that goes on. So it's not just, here's your medication, good luck to you. It's very much supervised. It's very much monitored on a regular basis, and allows the patients to have contact with the physician and or nurse every 2 weeks. Based upon that, with the larger visits being monthly. So it's a good setup, it's a good situation, and that's during the double-blind period. And then at the end of the trial, and that's what EOT, then they'll go into the open label extension. So this is an opportunity where they can, everyone will be on drug, everyone will know that they're on drug. And that's going to be for another 48 weeks, but we'll ensure that as patients are responding and doing well on the medication, that they'll continue to be able to have access to the medication until it's available on the market. So this allows us to really create two things: the ease and convenience of where would you like to be seen in the trial, and then secondly, how do we continue to maintain the medication if the patient's doing well on it? And our drug has some different qualities to it that are different than what we've seen with other drugs. And I'll show that here in a minute. But one of the biggest ones is there's no titration involved. So this means that typical sodium channel drugs would have to be titrated over a period of time, a lot of times 6 to 12 weeks, before you could get to what would be a stable therapeutic dose or get to a dose that would actually begin to have benefit. So what we've done is, because the molecule is not like what we've seen before, it's been designed differently with different qualities and properties, and we don't have to titrate it. Meaning we can start immediately, and then the patients are able to be at therapeutic levels, on the first dose and able to maintain that. And the half-life is pretty long, so that means it's once daily dosing. And it's also then J-tube and G-tube friendly and keto friendly. So this is really a molecule that's designed for the kids versus what we've seen historically as drugs are kind of reverse engineered, if you will. They take adult products and kind of figure out where they might work in the children. These are designed for the children from the beginning. So we're excited about that. And so that gets into then other innovative ideas within the trial. So not only can you do it at home, but what we've done is we had set up this trial to be approximately 20 patients. In this case, we had 16, and they were split into two groups, and one would be Relutragine, so this would be the active therapy, and the other one would be placebo, but placebo only for 4 weeks in the 16. So this is also a different trial design. Most of the time you would have active treatment and then you would have placebo for the entire time. And we already know what happens if we don't treat the patients. We already understand what that's going to look like. So what we wanted to do is create an opportunity for us to have a placebo-controlled trial, but in a way that allows this group to have drug for 75% of the time. So that means they would only be on for 4 weeks during sometime during the 16-week period. So it was a new idea. I really encourage the company to celebrate that, to be innovative, to be different, and to design this in a way that is beneficial to the kids. I think was great. Then they go into the open label Relutragine. Our key endpoints that we looked at were adverse events. We wanted to make sure this is safe first and foremost. And that's the thing that's near and dear to my heart, is I want to make sure that the kids, just like my child, would be protected and be safe during the course of the trial. And then we can begin to look at the efficacy. So how well did it help with the seizures? How well did it help control or reduce the number? But also, were there periods where there were no seizures at all? And so this helps to elevate the idea of treatment isn't, maybe there's a new chapter we can write, if you will, right? Seizure frequency goes down, but can we get where it goes away altogether? That should be, I think, the goal that we look at in treatment and developing new drugs. And we did introduce here a clinical and caregiver global impression of severity and improvement. So what this means is at baseline, we look at where would you rate your child and where would the clinician rate your child as far as severity. And then we can look at the end of the trial after treatments, see if anything changed. In addition to the seizures, in addition to the seizure frequency and adverse events, did anything change about the child and the function? Because this is important, I think, from what I've heard from the families that I've spoken to and been at different meetings, this is like one of those really important things, is what else do we get with this beyond just seizure control? So when we look at the demographics, and this kind of helps families to get an idea of what did the patients look like that were in the trial. So on average, they were about 6 years old. And so when you look at it, it was split evenly in this population between 8A and 2A patients. And then we look at the age of onset of seizures, and it can be different. The 2A population for the gain of function typically is the onset of seizures within the first 3 months of life. They come out very early with seizures. And we looked at the patients and the number of anti-seizure medications that they're on. So you can tell from these numbers that there were a lot of medications that the kids were on. So these are not just like very, mild patients who are treated with one medication and everything seems to be good. You had majority of them actually were on 3 to 4 anti-seizure meds and still not getting adequate control. So I think that's an important component to note. When you look at the baseline seizure frequencies, so in these groups, you're right about 55 seizures per month. So this is a very active group of patients that we're looking at, and relatively severe in this case. When we look at the safety, because I think it's always important to look at what did the drug do from a safety standpoint. What we saw, and again, keeping in mind the placebo group was a limited period of time in which they were, so only 4 weeks during that 16 were they on placebo. What we saw is there really wasn't an increase in risk of infections. These type of things, I think, come naturally with children. We go through seasons here where we get sick periodically. So these are things that do tend to happen, but there wasn't anything that really stood out within this as being a major issue related to study drug. And so what we saw, most of the adverse events were mild to moderate, no drug-related SAEs, and no dose reduction of relutragine was required. So that's an important piece that indicates to us it's well tolerated and that the patients did do well while taking the medication. So when we look at the safety being established, then we look at what did we do? Did we actually create benefit? And to what extent was there benefit? So when we look at it in the patient population that was studied, we did see at the end of treatment, there was a 46% reduction in the placebo adjustment of motor seizures. So what this means is that the number dropped by nearly half during the double-blind period. And these were kids who had already been maximized on other treatments and had already been on other therapies. So they were coming into this was in addition to, which is really a strong signal to us that where other drugs had fallen short, we're able to pick that up and run with it and to create better outcomes, for these kids from a seizure standpoint. And I think the part of this that I think is really, really impressive to me is when we look at the number of patients who are able to get seizure free. So typically when I had spoken to parents in the past, they're always like, yes, we'd love to be seizure free, but there wasn't as much hope in their hearts, I think, sometimes that it would happen because it just seems to be more difficult. The kids tend to be more, fragile and with regards to seizures and breakthrough seizures and dosing. So what we saw is 33% of these patients in the trial did end up to be seizure free, and they ended up being seizure free for a long period of time. So this is impressive to us because you're always kind of waiting for the next one, when's it going to happen, sort of thing. And so then if you get periods where the seizures go away, not only did they go down significantly, but then they also went away in a significant number of patients in this case. It's really a strong signal to us that the drug is doing something different. So as I showed you before in the discovery phase to the preclinical phase to the healthy volunteer phase, the drug is actually acting, as we would like it to, and it's acting differently than what the other sodium channel drugs have done. And then when we look at it in the patient population and the 2A and 8A patients, we did see that the 2A and 8A patients got better. And so this is good because this is in line with what we would expect to see. But very much, I think, higher magnitude of patients were able to get seizure freedom than what we've seen with other molecules historically. So this is, for us, very exciting. And then, as I mentioned before, when we look at the caregiver and clinician-rated global impression of change, and this is important because it helps us to look at what else do we get beyond just seizure freedom or seizure control. What we saw is improvement in disruptive behavior. So as rated, both the dark line is clinician rated, so physicians were rating these, and then the caregiver was rating the light blue ones. And so what we saw is disruptive behavior got better. The families noticed that the kids were better behaved. Communication improved as well. And so one thing I always stress is communication isn't necessarily only verbal communication, so it's not necessarily speech only, but the ability to act, to react to the environment and interact with the environment around you is an important component of communication. So we're seeing these developmental changes occurring. Seizure severity and intensity did go down, as you would expect. As the medication is working, you did see the seizure severity go down. You saw the intensity go down. And ultimately, in long term, it did go away for those 33% and drastically down for the others. But what was interesting is usually this alertness will kind of go the opposite direction. So you tend to, when you control seizures and with the old medications, it almost creates more of a sedating effect. And so some of the families may have seen this with different medications where like the seizure's gone or the seizure's getting better, but my child isn't as interactive with the environment around them. What we saw with this is as clinicians rated these kids, they saw 69% of these kids increased in alertness. So in addition to the reduction in seizures, they were able to interact much better, able to function in the environment much better, which I think is a very strong signal to me that we have something else going on, that there's an additional benefit beyond just the seizure control. And so this is, for me, exciting. And I hope it's exciting for the families too, because it's kind of what we've been looking for a while. when you look at the total summary of what we have, as mentioned before, the 46% placebo-adjusted reduction is impressive. And I mentioned long term. So when we look at this at a longer period of time, and as the patients go into the open label extension, we see that 46 turn into 75. We've never seen something like that before. I've worked on other molecules. We haven't seen that, and especially in this population that is much more, in my opinion, challenging to treat. We haven't seen a reduction of 75%, and I haven't really seen it in other treatments as well. So this is exciting because this is again what we want to see. We want to see families get better. We want to see kids get better and function better. And I think that's what we're seeing in these data right now. And this is also in a population that have been treated with a lot of medications. So as mentioned before, a lot of them were on, 3, 4, 5, anti-seizure medications. And then you add this on. So this then becomes the fourth, fifth, or sixth medication for them. And so you would expect to see the opposite with regard to the function. You would expect to see more sedation, but this one definitely is not sedating, which I think is a huge, huge opportunity for us to look at clinically and outcomes for these patients. And then the seizure control, the seizure freedom, and the duration. So two things, 33% getting the seizure control and then being able to maintain that for longer than 28 days is really encouraging to us, which allows us then as a company and as a group of parents to say, hey, I do think we have something special here. And I think we have an opportunity to help other families, and we have an opportunity to help other patients who are really struggling with what they're dealing with, and to look for different endpoints. How do we move this forward in a way that as families we expect more and that we can do more, with these better medications. And again, some of the ones we've used are very old, and our expectations for outcomes are not always the best because the medications just don't function the way we'd like. So this is a signal to us that we really have something special, that I think is beneficial to the families. And we have an expanded registration cohort. So going back to that diagram where we had all of these different hurdles along the way, typically when you go from phase 2 into phase 3, there's a break that occurs, right? So you have to retool the sites. You've got like more time involved. But for us, it's in our DNA to try to move as quickly and innovatively as quick as possible. And so what we see here is that we're able to take and expand this trial from this cohort to these 2 additional cohorts that can be added, which allow us to get to registration phases with the different regulatory agencies. So what this means is all the participation that the families have done, and by them being in these first parts of the trial, and then more families signing up to be in the second part of the trial, this actually helps the entire community. This helps the community be able to get medications to market more quickly, and it helps it to do it in a way that will create more treatment options. So families coming through will have again an opportunity to get more cutting edge medications and have an opportunity to have earlier control of certain conditions. So we're excited about that. I want to say thank you to the families for sure. The families who had been part of this and who had been willing to sign up. I know sometimes it can be frightening to be in a clinical trial because you're not really sure what happens. What we see is these data are reassuring because we're seeing it across each of the models and each of the different phases that we're seeing that the molecule is very safe, well tolerated, more so than what we've seen historically. And I think that's an important component, is that the drug works effectively. It works in a very precise manner. It's very selective in how it works, which is good because that's going to eliminate some of the side effects and tolerability issues that we've seen with the other molecules. And really, I think it has a chance to change the landscape of, the way we treat DEEs in general. So that meant, kind of within that segue, it helps us to see, where we're looking at. We're trying to get this trial enrolled and trying to get the additional families in there to balance out the cohort so that we can do the analysis and determine that the drug is efficacious and safe. Again, the safety component we've established, we want to make sure that it's efficacious for the additional registrational pieces. And so that's where we are now is it's open for enrollment. So families can join anytime that they're interested. There's a QR code in the upper corner that they can scan. It'll take them to the website. And what we have set up is kind of also unique and novel in that there, if you go to emboldstudy.org, then you're able to go to get into the queue for the nurse navigator, and the nurse will actually talk with the families. It helps us to understand where the families are, their level of interest, and are there ways that we can help facilitate them being in the trial. So that may mean, if people are willing to relocate to certain areas for whatever reason, then it gives them an opportunity to make sure that we know where they are and see what we can do to help them. Is there something that Praxis can do to help with either relocation or any challenges that they might face? And we do that on a case-by-case basis. So that's really the first step, is to get into emboldstudy.org. Sign up for Nurse Navigator, have a discussion with the nurse who is not a Praxis employee, so a completely independent, completely private with regard to health information. And then we can figure out, how to best help the families and really provide what they need when they need it. But the trial is gonna, here's the design of the trial, obviously, or I mean, here's the timeline for the trial. We're hoping to get this enrolled quickly, to be able to get to top line, to get it, to regulatory agencies for review. And it's the same design. So Relutragine for 16 weeks or placebo for 4 plus Relutragine for 12 weeks. And so this 4 weeks is anywhere within that 16 weeks for these patients. So we're excited. And sometimes the question comes up, like, who is the patient that we're looking for? The patient is going to be someone with a diagnosis of SCN2A DEs, which is supported by the clinical and genetic findings, has 8 motor seizures per month. And so they're looking at it in the 4 weeks prior to screening and the 4 weeks at baseline. And then the 2 to 18 years of age currently. And in the original cohorts, we had limited to 1 sodium channel blocker. But in the new study, in the new cohorts, we've actually expanded that to 2 sodium channel blockers. Looking at the safety and tolerability that we noticed, we do believe that there would be no issues with the patients taking 2 sodium channel blockers, plus then adding Relutragine to that treatment regimen. So this is huge because I think it creates more flexibility for the families. And ultimately, then when we look at kind of the summary of where this molecule sits and what the data have shown us is well tolerated, in both the pediatric patients, but not only tolerability, but efficacy. We do see the signal for efficacy very strongly for both seizure freedom as well as in seizure control. No need for titration, as I mentioned before, so patients can start immediately on a dose that would be efficacious for them, and they can maintain that dose. And the long half-life means that they're not as susceptible to drug fluctuations. So when you think about it, half-life is how quickly a drug is eliminated from your body. This drug will stay around for a while, which means that it's not going to go up and down. So once daily means that the drug is pretty stable, and has a longer half-life, which I think is good. At my house, we're on drugs that are twice daily. And so then we have to make sure we don't miss a dose and those type of things, because those drugs eliminate very quickly. This one will stay in the system to create that additional benefit and health opportunity for the patients to be able to maintain their seizure control. And as mentioned before, that's where the once daily is. And it's designed specifically, to be oral or J-tube or G-tube administered, as well as then keto diet friendly. Because again, these were designed for the children. So that's another major difference, is the molecules are created specifically for the 2A and 8A communities, and specifically for pediatric use. So we're excited. Like, the data have been, I don't know, as much as you can get excited about nerdy things, like the data have been very encouraging, because it tells us that when we leverage what we know and we leverage our past histories and what we've learned along the way, that we can develop better drugs faster, and we can do it in ways that haven't been done before, and be innovative and really, push, because again, it makes a difference to the families. And if it makes a difference to the families, it makes a difference to us. So we want to make sure that we can support them in any way, shape, or form. So with that, I'd like to say thank you. Thank you for the opportunity to address the families, and hopefully if there's questions or needs that they have, by all means, they can reach out to us at any point in time. The nurse navigator at the emboldstudy.org is going to be the best opportunity to get into the queue, and then we can look at the case by case of what needs to be done to help the families.